So thank you everyone today for joining us for our live webinar, which will be presented by Anna Nicolaou from University of Manchester, entitled Bioactive Lipids, Mediating the Functionality of the Epidermal Barrier. As usual, the webinar is being recorded and we'll have it uploaded onto our website and our YouTube channel within the next couple of days. It's going to be around 45 minutes of talk and then maybe 10-15 minutes at the end for questions. And as usual, if you could post your questions in the chat or in the Q&A during the talk, don't wait till the end. Do it during the talk as they come to mind, because what happens then is I'll take you in turn and unmute you and you can ask your own questions. So first come, first served, get your questions down. So now I want to introduce Anna. Anna did her BSc and PhD in chemistry at the University of Athens, Greece, and then moved to train as a postdoctoral fellow at the School of Pharmacy, University of London, and spent time studying enzymology, lipid biochemistry, and lipidomics in Paris, Marseille, and Boston. She then moved to the University of Bradford in '97 and was awarded a personal chair in 2008, and then moved to her current post at the University of Manchester in 2013, where she's now Professor of Biological Chemistry at the Faculty of Medical and Human Sciences. Anna's research focuses on bioactive lipids and mass spectrometry-based lipidomics, with emphasis on the molecular mechanisms that mediate the role of eicosanoids, endocannabinoids, ceramides, and related mediators in inflammation, immunity, cellular communication, and tissue responses. She's got a long-standing interest in skin inflammation and the role of lipids in acute and chronic cutaneous disorders, the involvement of fatty acids and their metabolites in cancer, neuroinflammation, reproductive tissues, and cardiovascular systems also of interest. And her work's been supported by research councils, charities, and industry funding. Anna is a fellow of the Royal Society of Chemistry. She is or was on several boards, including currently the ISFOL and also Eurofed Lipid, prostaglandins, leukotrienes, and essential fatty acids, and BBA, molecular cell biology of lipids. And with that introduction, I'm going to hand over to you, Anna. Look forward to very much to hearing your talk.
Thanks, Val. Thank you so very much. And thank you all for joining us today. So what I'd like to do is I want to take you through some of the work we've been doing here in Manchester on trying to understand how lipids, a variety of lipids, mediate the functionality of the barrier, both in terms of making the barrier function and making the barrier what it is and supporting its functionality. So let me make a start. Let me make a start by kind of reminding you why we bother to do all that. Skin is something we see first thing in the mirror. Skin is something we're very aware of. And skin conditions or systemic disease that has a skin involvement becomes very obvious to us. And you can see on the screen pictures of skin that looks unwell when inflammation and impaired barrier functionality is underlying all these conditions. And these conditions could be diseases like psoriasis, like acne, but also it could be a normal process like wound healing that may or may have not turned up well. And also the barrier and inflammation that can affect it is also impacted quite a lot by our exposure to environmental factors. And I have here some pictures of what happens to our skin if we overdo it in terms of sunlight exposure. So that's the big why we trouble ourselves and our volunteers for all these years to work on skin. And this is how skin works, really. I tried to put in a little cartoon the functionality, the different functionalities of the skin barrier. The barrier is there to protect us from the environment and also keep vital instruments safe. So we have, my cursor is here, thank you. So we have the permeability barrier that stops excessive water loss from us and stops small molecules to come in. We have the chemical barrier on the surface that allows the microbiome to live and thrive under healthy, normal conditions and totally in control. It stops altogether these barriers. They stop chemicals, irritants, allergens to get through the skin. And of course, we have the collaboration of the immunological barrier with aspects of innate and adaptive immunity. So the collaboration of the different layers of the skin are behind this functionality. Now, where on earth would lipids come into this? Lipids are an integral part of skin. We have lipids covering the surface of the skin. We have lipids, a very particular and unique combination of lipids forming the stratum corneum, the top of the epidermal barrier. And we have lipids allowing the keratinocytes, the fibroblasts, the immune cells to communicate and interact between them in the most classical sense of the way, the structural lipids, the lipids of the membrane, the lipids mediators. So all these organization, very detailed organization, and the balance of all these functionalities are behind the physical and immunological barrier. So over the years, and maybe you have heard me talking about this platform before, we've developed a quite extensive lipidomics platform. We can work with different tissues, different samples. We harvest lipids from the surface of the skin, and you can see here a sticky tape. So this is a tape strip that we use to harvest cells of the stratum corneum. We can use a sabutape to get sebaceous lipids. We can raise blisters and collect the fluid that is rich in mediators, protein and lipid mediators of inflammation. Or we can take biopsies from treated or untreated skin and work on the epidermis and dermis. We work in across 3 different platforms. When we know exactly our question and we have standards and calibration lines for the metabolites of interest, we tend to follow a targeted lipidomics approach using UPLC and a tandem mass spectrometer. When we have more broad questions, so we're looking at changes in lipid classes that we're not in a position to understand exactly the members of the group, how many, or we have a very small number of internal standards to use with. We follow an untargeted approach, and for this we have a supercritical fluid chromatography module connected to time-of-flight mass spectrometers. And of course, work has progressed quite a lot in terms of lipid imaging, using targeted and untargeted approaches and in situ lipidomics. And this is an exercise that we followed in collaboration with Waters here in Manchester. So from those 3 approaches today, I'm going to follow technologies behind the first 2 approaches. And let me go back to the barrier. So this cartoon summarizes, I want to hope, the functionalities of the barrier from the point of view of the lipids. So you see the skin in the middle, and you see what can happen to it as the environment tries to upset it, or water is lost because of disease and broken barrier. We have the physical barrier, the stratum corneum and the underlying lipid layers. We have the immunological barrier, stratum corneum and the viable parts of the epidermis with the key immune cells, Langerhans cells, keratinocytes and T cells. We have the microbiological barrier where we have bacteria, fungi, viruses on the surface of the skin. And to make a start, I'd like to take you through the way I see and understand the physical barrier. Now, to understand the physical barrier, we need to go take a step back and look at lipid changes during the epidermal differentiation. And in case you haven't done any work with epidermal keratinocytes before, as the keratinocytes move from the lower viable layers of the epidermis to the top to form the stratum corneum, they differentiate and they slowly lose their viability, if I can call it this way. They become flat keratinized cells that form the stratum corneum. And this is what is actually happening. The stratum corneum is, let me start from the top. Imagine this structure here, in between 2 keratinocytes, at the top, those yellow structures that have been keratinized and now they're called corneocytes. We have an envelope of proteins that surround them, and on this envelope we have ceramides attached to them, And in between those two structures, we have a variety of lipids. And these lipids are very particular to the stratum corneum. We have free fatty acids, we have cholesterol, and we have ceramides. Acyl ceramides, very long ceramide, you will see the structures later on, and omega hydroxy ceramides. Where are they coming from? They're coming from the lamellar bodies of a proliferating keratinocyte. As it becomes differentiated, they are excreted, and the lipids of the lamellar bodies, they're hydrolyzed to give you these lipids that are in between the layers of the corneocytes. Where do they come from? They're synthesized at the Golgi apparatus, going back to the endoplasmic reticulum, and for this we import fatty acids, and there's a synthesis pathway that leads to the different ceramides we see. What is happening with the stratum corneum, and why is it so important to understand it? A lot of skin diseases demonstrate lipid dysregulation. We have changes in the abundance and quality of the ceramides of the stratum corneum, the fatty acids change, and all these changes, they tend to be associated with poor functionality. And this is something we see quite a lot in aged skin. So this is my introduction to take you through some of the work that has been keeping us busy for a few years now in Manchester. As you very well know, and you don't need me to repeat it, but I will, we are living in a period of affluence, and we live longer, and it is estimated that by 2050, 1 in 6 people in the world will be over 65 years old. So we have an ever aging population, and that is really good news, provided we maintain good quality of life and health as we grow older. The problem we have with the skin is that skin health declines with age, and as people get older, they lose functional capacity, they may lose their independence, so all sorts of skin-related problems appear, and it would be really good if we could find ways to fortify, to strengthen the aged skin, to be able to live longer, but also in a good health. And when we started this project, we got the opportunity to be involved in a menopause study that was happening in the lab of collaborators here in Manchester. And our friends and collaborators looked into menopause because in women, we have this biological, I don't want to call it problem, it's part of life, right? The moment you hit the age of approximately 45, menopause comes in, and the changes we see during the menopause are part of the chronological aging in females. And it is considered now that menopause is the first clinically significant manifestation of skin aging. And together with the declining levels of estrogen, there's a reduced skin strength and elasticity, increased dryness, there's a change in the immune profile. So a lot of things that are associated with aging. So it's a good model. And when we got biopsies from a cohort of women before and after the menopause, so these are premenopausal, postmenopausal women, we saw that there was a change in the thickness of stratum corneum and the viable epidermis. And that led postmenopause to a thicker stratum corneum. And what we also noticed from that study was that women in HRT, hormonal replacement treatment, their skin appeared to partially normalize during this treatment. So we had a closer look at what was happening in terms of lipids. And again, working with biopsies, we saw that there was a decline in ceramides. Now, biopsies, in working with biopsies, we had not purely epidermis or stratum corneum. We had the live part of the epidermis. We also had some dermis possibly. So we decided to look more carefully in what is happening in ceramides. So I have this slide here just to remind you of the complexity of the ceramides in the epidermis. So this is the de novo biosynthesis that leads to the formation of dihydroceramides. And this is a critical step, is the ceramide synthase that takes the sphingosine base and acyl chain, a fatty acid, and links them together to form this precursor dihydroceramide that in the skin, following desaturases reactions catalyzed by some known and some unknown desaturases, we have a range of different ceramides. And these are some of them. Today we know of at least 21 classes of ceramides, including more than 1,200 individual ceramide species. And in every case, we have a combination of a sphingosine base and an acyl chain. And these are only 4 sphingosine bases and 3 combinations of acyl chains. But we also have not only 3 ceramides like the ones you see on my screen, but we also have protein-bound ceramides. And we have not only the simple ceramides you find in other parts of the body, but we also have these acyl ceramides that carry linoleic acid as terrified at, it's the omega-linked, unique to skin hydroxyserum, acyl ceramides that carry linoleic acid, one of the essential fatty acids, a key fatty acid for skin integrity. So this is some of the data we get when we analyze ceramides of the stratum corneum. These are some of the classes of ceramides we're going after. And if I take one at random, for example, this slide here at the top, you see the structure of the ceramide, and you see that each dot represents a different species. And you can see how they vary depending on the number of carbon on the sphingosine base, and the retention times change. So they come out in waves in the mass spectrometer, and it's really a pain to look for them. We have to type in manually more than 900 transitions, and I'm glad I'm not working in the lab anymore. But it is what it is, and we do what we have to do. And this is the kind of approach we follow to get this data. If this is a sample of a skin biopsy or skin on somebody's body, actually, we use these little tapes, sticky tapes, to take layers of the stratum corneum. And if you remove more or less 20-25 layers, you can reach the bottom of the stratum corneum. And we did a pilot study going up down to a depth of almost 15 tapes. We grouped them in bundles of 5. We analyzed the ceramides because we wanted to see how many tapes we needed to have from each volunteer in order to get represented data that we could rely upon. And we decided to use the 5 top layers as they were giving us a quite good representative measure of the cutaneous ceramides. And this is what we found in the menopause. This slide shows you in a snapshot what is happening once you hit the menopause, the blue bar. Almost all ceramide classes, the most abundant ones here and the less abundant ones there, they're going down. And luckily, women in HRT, their skin is able to recover from this change. And you see the orange bars going up. So we wanted to find out a bit more about how these things are happening. And we organized the ceramides in terms of average carbon number. So we can appreciate where in this molecule, this typical ceramide, the biochemistry changes. So these are all the average carbon number if we take all the carbons of this structure. But if we consider only the carbons of the base, the blue part of the molecule, the blue sphingosine base, you see that we start seeing the same changes. And we do not see that if we examine the acyl, the average acyl chain length, the red part of the molecule. So this experiment showed us very clearly that the changes come from the sphingosine base. And we looked a bit into some of the precursors of cutaneous ceramides. And this is an experiment we did analyzing sphingomyelins from the same samples, where we saw a decrease in the average carbon number as you can see here, but we saw an increase in the abundance of these shorter sphingomyelins. So the menopausal, postmenopausal skin is going through some changes in terms of how it produces and how it stores these ceramides. And when we started looking at correlations to understand a bit more of the physiology behind this change, we see a negative correlation with transepidermal water loss. So the shorter our ceramides, the less efficient the barrier is, the more water it is getting lost. But we also see a good correlation with estradiol in serum. So getting some primary human keratinocytes that we grew and differentiated in the lab, and we measured NS and NDS ceramides. These are the two classes that are produced in culture by these cells. We saw that estradiol could reverse the change. So this is now a question we want to explore a bit more and understand where exactly estradiol works in this respect. So here's what we learned from the menopause study. Postmenopausal skin has less abundant and shorter ceramides. Potentially, this may be coming from the top of the de novo biosynthetic pathway, the palmitoyl transferase, serine palmitoyl transferase, that is the rate-limiting step of the pathway. These changes could be indicative of barrier insufficiency. It could explain to some extent the dryness and the irritation and the impaired function we start to see in postmenopausal skin. And there's a direct link, we think, between estrogen and ceramides, and that is subject for further exploration as the lab continues to work on this topic. But the question now is, okay, this is what is happening in females post-menopause. Can we extrapolate the same thing in terms of males? And what is happening as we grow even older? So at the same study we were doing, the menopause study, we were recruiting for the study that I'll show you some of the newest data from just now. So we recruited males and females into age groups. 18 to 40, the younger ones, and plus 70, they are very old individuals. And we looked at changes in an oily site, the forehead, in a moist site, the armpit, and a dry photoprotected site, and for that we used the upper buttock. And we used tape strips to harvest lipids. We took swabs to get microorganisms. We took some biopsies for aging markers, and we measure also transepidermal water loss and pH to get some functional data. So in terms of clinical changes, we saw what the textbooks kind of say. As we get older, the pH of the skin is going higher. That has implications for skin biochemistry and the microbiome that lives on our skin, the microbiota. And you can see here in all sides, we saw this change in pH. Now having more material, and because we designed the study this way to explore a bit better the changes in the stratum corneum, we looked in an untargeted approach. We looked for triglycerides, disacglycerols, and free fatty acids. And these two complex lipids, they tend to come from sebum, while the free fatty acids are a component of the stratum corneum lipids. And we saw that in aged skin, we do have lower levels of triglycerides. And there's a negative correlation with the skin pH. We also saw, I'll skip the slide, hang on a minute, there you go. Disacglycerols were also lower. And free fatty acids were also lower. So as we age, key components, key lipids of the stratum corneum and the epidermis are getting lower in terms of abundance. But we did not see any changes in cholesterol. We saw changes in the abundance of ceramides with age. They decreased. And what is, I think, quite telling is not the quantity so much as it is the quality. If you look at these slides, we have a lower average carbon number of these ceramides. We have shorter ceramides, both in males and females. And what I quite like in some respect is that all the males, they seem to be losing the acyl ceramides more than females. We're still working on this, and it could be that the females will show the same characteristic, but it starts to show that it is possible males and females have changes, have some differential changes in the stratum corneum lipids as we grow older. So what we have learned so far, yes, we do have profound changes in female stratum corneum lipids, and that could be attributed, amongst other things, in hormonal changes. And we also have changes in sebum lipids, triglycerides, disacglycerols. And it is possible that it's not only the global changes we see as less sebum is produced, but also the quality of the sebum. And that is, again, a question we are following. What's the potential impact of these findings for aged skin? Possibly an impaired functionality and integrity. And by that, it's not, I'm not saying that it's unhealthy, the aged skin, but it could be that it is not easy for aged skin to deal with challenges. And the quite important finding, I think, for me is that these changes reminded all of us to the changes we see in inflammatory skin conditions. It could be that this less robust aged skin could be more susceptible to pathogens, to chemicals, to UVR, to water loss. And something that is, I think, quite important to follow, and we do have followed it, and I'll share some data in a little while, is the importance, the implication of all this for the microbiome. The microbiota, the profile of microorganisms we see on the skin, they rely upon sebaceous and other lipids for nutrition, but also for generation of antimicrobial agents. So it is quite possible that these properties of the aged skin reflect negatively on the microbiota that live on our skin. And I think that brings me to examining the microbiological barrier. Let me remind you of how we consider this barrier is a cocktail of microorganisms and also the qualities of the skin, because we have a relatively acidic pH that is part of what is usually referred to as the acid mantle. It's an environment that maintains a fine balance between pathogens and commensals, and everything is controlled with antimicrobial lipids, antimicrobial peptides, etc. So let's have a proper look at it. This is a classic slide everybody in microbiology is using by the seminal review by Grice in 2011, and it shows you where in our skin how we have different types of microorganisms. It shows you how different microorganisms prefer different environments. So we have a different balance in the oily sides or the moist sides or the dry sides of our skin. And you can see here some of the bacteria and the fungi that live on our skin. And malassezia is something that maybe it's more familiar to all of us because of its implication to dandruff. And we have the propionibacteria or Corynebacteria and staphylococci, some of the key bacteria that live on different areas of our skin. So what's the story with lipids and the microbiome, and the microbiota, I should say? There is a relationship between the two. It's a dynamic relationship. Let's start with the epidermal lipids. In our the skin or the surface of our skin, we have sebum that has triglycerides, fatty acids, and it's good quality food for certain microorganisms. The pH of the skin is regulating the functionality of some of these microorganisms and is attributed in part to the presence of free fatty acids. We also have antimicrobial lipids, sapienic acid, palmitoleic acid, sphingosine, the free sphingosine base. They can all control the growth of different microorganisms. At the same time, the microorganisms that are on the surface of the skin, they utilize triglycerides, for example, as nutrients, but they also produce antimicrobials, and they keep on check other microorganisms also on the skin. Then we have the issue of oxylipins, short-chain fatty acids that are produced by microorganisms on the surface of the skin, and that could be irritating, that could call in immune cells. We also have ceramides that are produced by these microorganisms, and they can affect keratinocyte differentiation. So as you can see, it is a very dynamic relationship. And we wanted to see how this looks like in our aging study. So I will just share some data from the aging study. I'll focus on female skin and on the oily side where we saw quite clear differences. And as you can see on this side of the diagram, I have the microorganisms the bacteria mainly we see in younger skin, and on this side on the older skin. And it becomes straight away very, very profound that we have lost the blue ones. So we are losing a lot of microorganisms that are using in the younger skin, and they are not as profound in the older skin. And we see an increased diversity in the older skin. And being all our questions stem out of our interest in lipids, we wanted to see what is their relationship. So we asked the data scientists that support the team to look into a relationship between epidermal lipids and different taxa of the bacteria. And it becomes evident from the findings we have that lipid species can predict the prevalence of these microorganisms. And I have here some data from a lasso regression analysis, a model that tells us that certain diacylglycerols or certain ceramides and triglyceride species are better predicting the prevalence of certain microorganisms. And when we look at how this relates to individual species, both in terms of microorganisms and lipids, you see those heat maps that shows you again that there's more activity in older females because of the increased diversity. So again, this is something that is of interest to us. But at the same time, we mustn't forget that bacteria are not the only microorganisms living on the skin. We have a lot of yeasts. So Malassezia species are a topic of interest of our collaborators in Singapore. And we've started looking into what comes out of these yeasts if we grow them away from the skin. And we tried to compare data that has been gathered from the surface by analyzing surface skin lipids compared to what Malassezia is producing in culture. And we see that we have quite a lot of linoleic acid metabolites, to a smaller extent some arachidonic acid metabolites. And again, this is a question that we want to explore more and try to understand what is really coming out of the microorganisms and what comes from the skin and what the relationship might be. Because this could easily lead to inflammation and irritation that is observed in skin conditions. So what have we learned so far from asking questions about the lipids and the microbiome? Firstly, we've learned that we still don't know what is if these changes are causative or a consequence of the changes. Is the aged skin altering the microbiome, or the microbiome is also contributed to the changes we see in the aged skin? Do we have these changes being reflected in all different body sites? what comes from them, what comes from us, and what is the consequence for the aged epidermis, and is that really a big impact, and how big is this impact for the immunological barrier, something that is directly linked to the microbiological barrier, because let's not forget the microbiota educates and informs the immune response in the skin. So we are now down here in the blue box, the immunological barrier. Langerhans cells, T cells that come in, infiltrate from the epidermis together with the resident cells. The keratinocytes, an immunocompetent type of cell, very important for the immune barrier of our skin. And this is more or less how things work at a more molecular level. So this cartoon is showing you how we envisage, how we imagine the interaction between cells. So that could be a keratinocyte or an immune cell that is producing lipids, and that could be a keratinocyte or an immune cell that receives lipids. And in this case, we're dealing with lipid mediators, all these lipids that come from polyunsaturated fatty acid metabolism, or eicosanoids, or octadecanoids, or related species. We have hydroxy fatty acids, we have endocannabinoids, both from the N-acetylethanolamine and monoacylglycerol groups, and we also have simpler sphingolipids like sphingosine, like smaller ceramides, sphingosine 1-phosphate. They are exported from the cell either through passive infusion or transporter-mediated systems. They bind to receptors on the surface of either keratinocytes or immune cells, for example, and they mediate a range of events. And you can see here some of these events listed. So we know that all these lipid mediators could have an influence in the growth, proliferation, differentiation of the keratinocyte or the fibroblast. They can inform the migration and recruitment of Langerhans cells and neutrophils, of T cells. So it's a rather complex picture. And I'll just show you some slides just to kind of remind you of how different activities these bioactive lipids, especially the ones that are derivatives of fatty acids, and how important could be for different aspects of the barrier. So in this slide here, I'm showing you how the N-acetylethanolamines that are found at quite high levels actually in human skin, and quite a lot of them. There's a variety of N-acetylethanolamines. How they tend to get upregulated when we irritate the barrier. This experiment is from some old work we did using UV and SLS, sodium lauryl sulfate, a detergent that used to be in shampoos, etc. And now I think it's found in very strong cleansing products. But it upregulates all the N-acetylethanolamines. And all these things are coming up once the barrier is disrupted. The barrier is also disrupted or upset if we expose our skin to UV. And this slide here, again, from a study we did a few years ago, shows that eicosanoids remain upregulated up to 2 weeks after UV exposure. So yes, you had a lovely holiday. Yes, you stayed by the swimming pool. Yes, you got burned, you shouldn't. The redness may have gone down, but for almost 2 weeks, your skin is fighting the change. And we started to see second waves of some of the eicosanoids coming up, and we see immune cells staying in the skin. And that could be possibly the reason for the immunosuppressive properties of UV. I mean, these are just early days in our thinking. So the reason I shared with you these findings is because I wanted to start asking the question and get your support in trying to come up with answers. How can we support the epidermal barrier? Because we do have to support the barrier. Being aged or skin or younger skin, we have to be able to protect it, because it's our defense against the environment. It's a really, really vital barrier for us. Typically, and one of the easiest way possibly to address this by topical treatments. And I'm sure you've seen in the market quite a lot of ceramide containing creams and emollients that are applied to the skin, and they temporarily rectify issues of the barrier. But we're looking for something more permanent, something that we want to give to the skin to nourish it, something from inside out. And we have looked into fatty acid supplementation to come up with such regimes, and also we have started to explore the gut-skin axis. And let me share you some of the work we've done and some thoughts, and that will be the last question I will trouble you with today. So why fatty acids? Omega-3, omega-6 fatty acids are really, really important in many aspects. They modify the membrane, and once there's a stimulus, this modified membrane can start release precursors of lipid mediators of the omega-3 and omega-6 pathways with potent pro and anti-inflammatory effects, pro-resolving, less resolving properties in terms of inflammation resolution. We also have a direct impact on gene expression through transcription factors, etc. But we also have physical changes, the altered membrane fluidity that may affect the structure and functionality of receptors. We have changes in membrane microdomains. So it's, again, a multifaceted effect. And when we supplemented volunteers with omega-3 fatty acids for almost 12 weeks, and we looked at the changes in the skin, we found that there was a different picture of how the epidermis and the dermis dealt with the omega-3 supplement. And while the dermis doesn't seem to be taking much attention of what's going on, the epidermis is changing the lipoxygenase generated products, metabolites, and also some of the endocannabinoids. And what the epidermis is doing is reflected more or less in the changes we see in plasma. The data I show here in these radar plots, they show global changes that we assessed using an iterative group analysis. So again, you can see here in the purple lines what was statistically significantly elevated, and these are predominantly the lipoxygenase products that are much more important for the epidermis. What we also saw was that N3 PUFA treated individuals, once they were challenged with UV, we saw an earlier infiltrate of CD4 and CD8 T cells. And we don't really know if this is a good thing or a bad thing. Is it creating a memory, or is it we have an earlier infiltration and the skin is possibly overreacting? It's something, again, that has generated more questions than answers. But one thing we know from work we've done with omega-3 fatty acids and T cells is the omega-3 fatty acids can change the motility and the migratory capacity of the cells. And you can see here T cells that in an animal model, in a murine model, that were treated with omega-3 fatty acids, and using a transendothelial migration assay, you see the reduced ability of these cells to move. And once we injected omega-3 treated T cells in recipient animals, again, you see the loss of these migratory capacity. So when we looked for reasons behind this, we saw that both EPA and DHA can alter the ratio F to G actin in terms, and we think that this is one of the reasons for reduced migration, as you can see here. And we went even further down to look at membrane microdomains, and we looked at changes that were induced following the EPA and DHA treatments. We saw changes in sphingomyelins, Ps and PCs, and we saw differential changes depending on the treatment and the type of fatty acid that appeared to be accumulating in the P fraction, for example, the PC fraction that preferred polyunsaturates versus P that preferred monounsaturates. So a part of looking more closely at omega-3 supplements, and that's something we're actively doing, we wanted to have a look at something that has been quite a lot discussed in the context of a holistic approach of our metabolism. We had a look at the gut-skin axis. This is a link that is well understood and established, and there's what the gut is producing because of the microbiota and the diet and the lifestyle is reflected on the skin, and again the skin feeds back. So all this cycle is maintained through changes in the innate and adaptive immunity and metabolites that are produced by either side. And we found I don't know if we were surprised or not, but we did find that some of the gut-derived short-chain fatty acids promote the integrity of the cutaneous barrier. And you can see here in this experiment where we used an animal of atopic dermatitis-like dysfunction, and we fed the animals diet or short-chain fatty acids. That butyrate was the most active one, and it changed the expression of genes that are key for skin ceramides, and then it changed the skin ceramides. And you can see here the NDS and the NS ceramides. So yes, there is a link between the gut and the skin. Short-chain fatty acids are of interest, but we have to be careful because when they are produced by the microbes on the skin, they actually irritants. So again, more questions than answers. So what shall we do next? I think we need to mine the cutaneous lipidome for biomarkers. I think we should pay a lot of attention in our understanding of the lipid networks behind skin reactions in different disease or systemic conditions with skin manifestations. It's important to have good quality and reliable in vitro and in silico models to study all these reactions because volunteer studies are very expensive. And I think we need to pay more attention in understanding the functionality of the microbiota that live on our skin and how they're affected or affecting skin quality. I want to say a massive thank you to everybody who's been working with me over the years. Firstly, the biggest thank you to all our volunteers who help us with the clinical studies. I want to say a massive thank you to Alex. He's been instrumental in all the lipid work in the lab, and Susie, a very talented postdoc behind some of the key clinical studies we've done over the years. Everybody else is on this slide together with our sponsors, and I want to extend my thanks to you for staying with me this late hour.
Thank you. That was great, Anna. Thank you. So there's several questions, so let's get going. Hong Yin.
Yep, I was just a question to Anna. Have you done the proteomic studies of the skin samples? Have you found those elongases goes down in your aging study? And one more question, maybe. So the ultra long chain fatty acid that present in the ceramides, are they tend to be saturated or monounsaturated or polyunsaturated?
Starting from the second question, if memory serves, they tend to be saturated. And the first question, skin proteomics, no, we haven't. We've done some work, but we couldn't get good quality quantitative proteomics. We haven't progressed with that. So we do RNA sequencing, but no proteins.
Okay. So do you see any difference in saturation of free fatty acids in the stratum corneum with age?
I think the changes we see are qualitative. I can't remember. To be frank with you, I can't remember. We see monounsaturates and saturates. We want to start looking at more, maybe the presence of branched fatty acids, but not polyunsaturates.
Okay, Alan Brash.
Anna, very impressive. I think you're keeping your mass specs very, very busy. My question was, you showed that menopause is associated with lower stratum corneum ceramides, yet there's a thicker stratum corneum. How do you explain or rationalize that barrier layer being thicker while the major component, the ceramides, are in a sort of lower trend? And what's the overall effect on transepidermal water loss and barrier function?
Okay, so immunohistochemistry, so the histology shows a thicker stratum corneum. The fact that we have shorter ceramides doesn't mean that they are less, if I can put, let me try to answer exactly.
So they're not less abundant, they're just shorter, is that right?
That is the case because we always normalize our data to the amount of protein we lift from the tape. So if we have more keratin, more cells, we would find more, but we normalize it always to that amount. It is the shortness of the ceramide.
Okay, got it, thanks.
And transepidermal loss, the changes we see, they're not enough for us to declare a malfunctioning barrier. We think that the barrier is functioning well. It's just using whatever ceramides are available to make sure it functions. It's more brittle. It doesn't, if it gets harmed, it can't fix itself easy enough to perform all the jobs it has to do. Hence the dryness and the irritation we see.
Thank you.
Okay, thank you. So Jason Rodenthal.
So I noticed there was a slide with very specific ceramide heat maps. It was right before the melasthesia slide, and it appeared that, I think it was comparing aged to young, youthful skin. It appeared that there were very specific species that were being regulated in aging. And that suggested to me there might be some gene-lipid relationships. So maybe like the gene was being downregulated, that caused these differences, or upregulated, whatever. So the question is two-part. Is it known what enzymes produce those specific lipids? And if so, have you gone to look at like the gene expression of those lipids or of those genes in those conditions?
Yes, these heat maps, they were not actually gene expressions. It was the relative abundance of microorganisms. So
Okay.
It's a heat map linking lipids and microorganisms that we found on the skin surface.
I see. Okay, so when you got more microorganisms or less microorganisms, then you get more of that specific lipid species.
Yeah.
Gotcha. Thank you.
I'm going to ask a question myself, or maybe it's a kind of an observation as well. It's interesting, these changes that we're seeing with age, and of course, young children who don't have any estrogen have probably got the most elastic skin of all. But I always think, like, we look at young children they don't have all the testosterone, all the estrogen, all the progesterone, and yet they're the healthiest of all.
That's why. Absolutely. I mean, and at the same time, men don't have the female hormones to that high extent, so their skin is perfectly okay. Yeah. So, I mean, I would agree with you. I think it's a balance of a lot of different factors. So, if estrogens have an effect, I'd like to know where exactly the target is and how things are organized in terms of skin biology, but there are also other things that are important for the production of ceramides. And our cells themselves, they don't function the same way when we're babies, when we're adults, etc. So we're actually we're recruiting volunteers now for another study. We're looking for males, females, same age groups, all taken from 30 onwards. And we haven't gone to babies, but I want to see males and females as they change as we get older, how the skin changes. So I don't get stuck with with actual or playing hormones for everything.
I mean, because I guess growth hormone or other, there's so many different hormones and how they change during aging that could be playing a role in the transcriptional regulation of these pathways. Isn't there, beyond these hormones specifically? And I mean, is there any research going on into, what's happening that goes beyond this? Because I think that research into aging tends to have a strong fixation on menopause, andropause, and not really consider other factors that might be involved.
That's quite true. And throughout our studies, we always get samples to look at indices of chronological aging. So we can see how skin age is independent of what we add to it. I mean, again, you have so many products, we put so many products on our skin. Sometimes it's difficult to get rid of them and get real readings. So yes,
that's really interesting. So we have a question from Kevin.
Hello, very nice talk. Do you have any information on the history of use of skincare or makeup products in these people and the effect on the skin and lipidome, especially considering that women typically use more of these products during their life than men? So this could be a confounder in this study.
That is correct. So we ask our volunteers to refrain from using any products before sampling. And I don't know if they do respect that or not. I want to hope yes. But at the end of the day, we also we collect cells from 3 locations in the body: the forehead, where they may or may not have used products, the armpit, and the lower part of buttocks. And I think that is the area that is the driest, photoprotected, and maybe not so well cared. We see quite clean changes from photoprotected, untreated buttock skin.
Okay.
So that's our secret weapon.
Okay, thank you.
Okay, and I've got one more thing I want to go back to, which I was really interested in your PGE2 data that you showed on with UV. And I think more and more there is evidence coming out about PGE2 being anti-inflammatory, whereas it's always been historically considered to be a pro-inflammatory icosanoid. So do you think it's playing some dual roles in the skin? Because clearly UV is bad. And if you've got a lot of PGE2, I could imagine how it would also contribute to the inflammatory response. So what is it kind of playing 2 different roles here, do you think? And
I don't know, we do have from various studies, we tend to see a second wave of prostaglandins and some heat is coming up as the skin is repairing itself. I don't know if it is the repair of the damage, or I don't really know why. I don't always think that prostaglandin E2 is the bad guy. Yes, it's very much linked to inflammation, right? And yes, we see it when the skin goes red and everything. But at the same time, I think it's trying to fix some of the damage because of its proliferative properties, etc. So I don't know is the answer, but we always keep an open mind when it comes to the waves of the different mediators. I mean, in wound healing, for example, take PGE out and the skin won't heal. So I think it's homeostatic. It's important as well as
it's possible its role evolves as healing process continues based on expression of the receptors that it interacts with, maybe. But yeah, interesting. Okay. Well, we've had loads of questions. We've got through them all now, Anna. So you'll be glad to know I can let you go. But thank you very much for coming today. That was great. Anyway, thank you very much, everyone, for coming today. Thank you, Anna, for a great talk. And we'll see everybody next month, hopefully. Bye now.